Good afternoon and welcome to this Chemistry World webinar. I'm Ben Valsler, Chemistry World's digital editor. My role is to introduce you to the software, introduce you to our speaker, and then hand over and let you enjoy the presentation and the software demonstration that we'll be getting today. Now, first of all, this is GoToWebinar that we're using as a platform for this. And one of the key things about GoToWebinar is that it allows you to interact with our speakers. So probably the most important bit of it is right down at the bottom of the GoToWebinar dashboard, where you will find a questions box or a chat box. So do just put any questions you have into that box at the bottom. We will then put them to our speaker at the end of the presentation. If you have any questions about webinars themselves or anything like that, you can either get them in there, or if it's something that can wait until after the webinar, then email us cwwebinars@rsc.org, and we'll be more than happy to get back to you and help. If you need to adjust your volume settings or that sort of thing, you can either use your general computer settings, or there is also an audio tab in GoToWebinar that will allow you to make the changes there that you need to make. So that's GoToWebinar, but what's today's topic? Well, today we're looking at Synthia retrosynthesis software for the chemical industry. We're seeing a live demonstration of Synthia, which is software that can make your time in the lab safer and more efficient. By the time we're done, in about an hour's time, we will have seen a new way to use retrosynthesis to accelerate your research. We'll see how to save time and money by avoiding patent infringement. We'll look at alternative pathways to avoid hazardous or reach-restricted chemistry, which can help protect your co-workers and the environment. We will look at how to generate new ideas and protect intellectual property by discovering previously unknown pathways and routes to the compounds you want to synthesize. And we'll look at what needs to be done to design pathways that lead back to commercial starting materials. We're doing all of that by looking at what Cynthia can do for us. Now, helping us to put this together has been Sigma. They are a regular Chemistry World webinar partner. We work with them to look at a variety of different things, and Cynthia is certainly one of their products that we find most interesting. They have been an excellent partner in that they have helped us find Emma Gardner, who is Technical Application Specialist in Cheminformatics Technologies for Sigma. She is responsible for the commercial license. She's responsible for the commercial licensing of Synthia. She's got a PhD in organic chemistry from Brown University in the US, and was a senior scientist at Vitae Industries, which is a pharmaceutical technology startup in Providence, Rhode Island. So she's done the legwork. She has the right sort of background, as many of you, I'm sure, will be in similar areas of chemistry. And she's now the right person to talk to us about Synthia. So get any questions you have in throughout the webinar into that questions box at the bottom. We will hand over to Emma now, who will be able to pick up very shortly. And then I will come back to you to find out a little bit more about you, and then to ask your questions to Emma a little bit later on. Emma, thank you ever so much for joining us.
Great, thanks, Ben, and thanks for having me. And we're really excited today to be able to show you Synthia, our retrosynthesis tool, and how you can use it in your research in the chemical industry. So just to get started, I do want to briefly introduce Synthia, which was actually a software originally known as Chematica. It was developed by an academic group led by Professor Bartosz Grabowski and his team of graduate students. And so it was originally under the name Chematica, and there were quite a few publications about this. So if you've seen any of those publications referencing Chematica, know that this is that same technology. We actually acquired the software back in 2017 and are continuing to work with Professor Grabowski as we're continuing to develop it and commercialize the software now. So to give you an idea of where Synthia is going to fit into your workflow, I have here just, a very general overview of what goes into developing new chemical products. And of course, at the early stages, there's a lot of work that goes into figuring out what compounds to make. And there are some in silico tools to help you with this target selection phase. And then once those targets are identified, it's passed off to these synthetic chemists to figure out how to make those molecules and what's going to be the best route. And up until now, this is still largely a fairly manual process of, looking through the literature and understanding the precedent of what chemistry has been done, and then kind of manually planning out a route before you're able to actually get into the lab and do that synthesis and hopefully eventually a scale up. And this can be a fairly iterative process of, trying something and then having to go back to the drawing board. And so some of the challenges in this small molecule pathway development, the first of which just being that most of us as human chemists can't remember every possible reaction, or at least maybe not quickly recall them, that's ever been published. And so we find ourselves, kind of falling back into our subset of comfortable chemistry that we're familiar with and have done before, and sometimes missing a really key transformation that we don't realize is possible. And so then, of course, we look into the literature to see what has been done before. Many times established routes may not be an option for various reasons. Perhaps we're looking for something that's actually different than what's been done before. And failure can really happen at any step, and certainly with a multi-step synthesis, this can be particularly costly, even when trying to reproduce a published route. And then, of course, trying to make sure that you are avoiding any hazardous or regulated chemistry, or looking for more green chemistry can be another challenge in limiting sort of your options for designing those paths. And then finally, just making sure that those routes that you are finding are going back to commercial building blocks so that you can actually, get your hands on the right starting materials, because otherwise that can obviously add additional steps or make that route not viable. And so these are all some things that Cynthia can really help with to be able to aid in this route planning phase. It's able to find multiple routes starting from a variety of different chemical starting materials for all of your targets. It's able to find, new routes for known molecules as well as, routes for unknown molecules. And of course, all of these paths do go back to commercially available building blocks so that it's something that you can get your hands on to start that chemistry. And all of this, of course, can help you save time and money and be able to generate new IP when you're finding those new routes. So Cynthia is a retrosynthetic design software. It uses network theory and modern high power computing, along with our database of expert chemical knowledge to be able to very quickly design these paths going back to your both known and novel targets. And what's really at the heart of Cynthia is our database of hand-coded chemistry rules. So this is a database of basically all of the chemical transformations that you would find in organic chemistry textbooks or that you see in the published literature. And all of these rules have been hand-coded by PhD level synthetic chemists, which allows those rules to take into account things like recognizing if there are any incompatible groups present, or recognizing if there are groups that need to be protected. It's also able to take into account stereo and regiochemistry, but these are written in sort of a general enough way that they can be applied to novel molecules or to new substrates that haven't necessarily been published before. And what I have shown on the right-hand side of the screen here is an example of what one of these hand-coded rules looks like. So we use this smiles and smarts notation to be able to translate the chemistry reaction information into something that's readable by the computer. And we do have a team of chemists that are continually updating the rules and adding new rules as new chemistry is published. And our database is up over around 95,000 rules, and we're continually adding to that. And it is somewhat of an art to really be able to write these hand-coded rules in a way that can account for all of those things. And how our team does this is they will start with a general transformation and then do an extensive literature search to make sure that they understand the full scope of that reaction, which allows them to map out this decision tree of where that reaction can or cannot be applied and what groups are compatible or incompatible with the reaction. Once this is mapped out, they're able to then start mapping it onto a SMARTS template and then finally filling in the code. And really depending on the complexity of the reaction and the coverage in the literature, an analysis like this can take as long as a few weeks to really code a single rule or often a family of rules to be able to fully describe that kind of transformation. So this is a fairly laborious approach that we're taking to hand coding the rules, but I hope to show you why we see that this is really important. So of course, there are perhaps maybe some more straightforward or less time-consuming ways to generate a rules database like this, for instance, using machine extraction of the literature to more automatically generate the rules database. And when the Grabowski group was first developing this tool, they actually started by trying to do that using machine extraction to automatically generate a rules database. But they very quickly found that this approach had a lot of problems with it. So anywhere just from the presence of human entry errors in the literature records that would then get propagated into the rules and into the chemistry being proposed, to the presence of cross-reactive functional groups or distant substituents, they very quickly realized that molecular context is key, and this is something that's really missed by the machine extracted rules. So we know as chemists that, you can't just look at the bonds that are being made or broken in a reaction, because other parts of the molecule can have a significant effect on the reaction. And this is something that's difficult for those automatically generated rules to be able to take into account. And so to explain what I mean by that a little bit more, I have an example here of a stereoselective alkylation reaction. And the reaction on top would be an example of that literature record. And what's on the bottom would be the rule generated using machine extraction from this literature record. So typically with machine extraction, it takes into account the reacting center and about 2 to 3 atoms away from that. And that's everything that I have shown here in yellow. And so what you might notice is missing from the machine extracted rule is actually that stereo-directing methoxy group. Because it's farther away from the reacting center through bonds, it's missed by the machine extracted rule. So then when we try to apply this rule to other scenarios, if we have very similar substrates to the original literature record, like what's shown in A, it is able to correctly predict the product of the reaction. However, as soon as we start adding other groups, for instance, this example in B, where now there's this cross-reactive nitro group, it's going to create acidic protons. So in those basic reaction conditions, you would not get the predicted product. And then in the example of C here, where now actually there is no stereo-directing group, but because the stereochemistry was extracted into the rule, it still would predict a single stereochemical product, even though in reality you would get a racemic mixture. So for this reason, we switched to hand coding the rules, which allows us to give all of this molecular context information to the rules so that it can look at the whole molecule more like a human chemist would. And how it does this is, so when a rule in Cynthia is applied to a reaction, the first thing that it does is it will then extract and exclude the reaction core so that it can look at the rest of the molecule to determine if there are any functional groups present that need to be protected, or if there are any incompatible functional groups. If there are those incompatible functional groups that can't be protected, it will say that this reaction is not viable. Or if there are functional groups that can be protected, now it's going to look at a matrix of protecting groups and select ones that are compatible with reaction conditions so that it can actually propose potential protecting groups that you can use for your synthesis. The other advantage of hand coding the rules is that it allows us to have fairly comprehensive coverage of the reaction space compared to using a machine learning approach to generate a tool like this. So, machine learning is, of course, a very popular topic and can be very powerful, but it's limited by the data sets that it uses to train the models. And especially with chemistry, you need very large data sets and very clean data sets to be able to train your model on. And so this works very well with very popular reactions that are published many, many times, where there's enough examples, but it tends to miss more specialized reactions, what we call these black swan reactions, that are of lower popularity. They aren't published as much, there's not as many records of them, but they can sometimes be really critical if you need to prepare, that kind of scaffold. And so, for instance, we have this cycloaddition reaction for preparing these Syn-1,3 amino alcohols that at the time we encoded a rule in Cynthia for it, there were only 128 references. And so this is something that's plenty of information for our human chemists to understand and map out the scope of the reaction, but is really kind of far below the current thresholds of what you need for machine learning. So the other piece of how Cynthia works is now, once we have all of these possible transformations that you can do, how do we figure out what are the best overall paths? And so if we think about, maybe there are 1,000 different reactions that could be applied at each step, and you have a 5-step synthesis. Now just the total number of possibilities based on those combinations is this humongous unmanageable number. And it would generate a tree that looks sort of like this ugly graph over here, which is something that, of course, none of us, no human chemist wants to or really could go through and evaluate all of those options by hand to figure out what's the best route. But this is something that Cynthia can do using a sophisticated maze navigating algorithm. And so, that complicated graph, kind of, if we break it down, it starts really with just a tree, where you start with the target that you're looking to make. And so this is what Cynthia does, is it takes that molecule and then it looks at just what are all of the retrosynthetic cuts it can do just going one step back. And then it's going to score all of these cuts. And then it only takes on the best scoring ones and eliminates those others, and then we'll expand those nodes to see what are all the ways we can make those molecules. And then it will do the same thing where it's scoring kind of the cut as well as the overall path, and eliminating any dead end directions, and continuing to expand those nodes until it finds whole paths that are now terminating in commercially available or known starting materials. And then it will score all of these overall paths and rank them so that it can show you basically the best paths first. But you can imagine that, what's considered really the best path is something that is fairly subjective, may not be the same thing even for the same molecule, if, for different projects or different chemists, depending on different criteria. So we have something in Cynthia called these scoring functions, which allows you to really customize how Cynthia is going to rank the paths and score those overall cuts in general. And we'll see this more when we get into the live demo, but there are really a lot of different ways that you can customize how Cynthia is going to rank your paths. And so, some of the things that it's going to take into account, of course, the number of steps in the route. It can take into account the cost of starting materials, and you can customize, how much you can set limits for that. It can also sort of direct it toward more protecting group free synthesis if you're looking for that, or if you don't mind using protecting groups. And then you can define what you want to use as your starting materials, whether you just want commercial compounds or known compounds. And you can also actually add your own internal inventory and have those as stopping points in Cynthia as well. But you can really customize it even further than that. So you can tell it if there's any starting materials, intermediates, reagents, or whole kinds of reactions that you don't want to use, you can give it that information, which allows you to, be able to find alternatives to toxic compounds if there's a route you're looking to avoid there. You can avoid whole lists of substances. So if you wanted to or need to avoid retro dysregulated substances, you can put that in and be able to kind of hide whole reaction classes and be able to promote, more green chemistry. So in this way, you can really kind of tailor Synthia so that it's finding, the best routes for you for that project. And so when we first saw this technology, of course, we were very interested in it. But at that time, everything was just on paper, and there really was no evidence that you could actually take the routes from Synthia and be able to execute them in the lab. And so we wanted to design that experiment to be able to test the routes. And so we took these 7 target molecules that I have shown on the screen here, and we had 6 of our chemists who were working on these projects coming from one of our 2 sites in the US, either the Cerulean site in Texas, which makes analytical reference materials, or our Sigma-Aldrich site in Massachusetts, which makes small molecule screening libraries, and had them actually travel to Poland, to the Polish Academy of Sciences, where the Grabowski group works, so that they could meet with the team and have face-to-face training so that they could use the software to plan the routes to these target molecules. And these were molecules that were of high financial interest to us, so real projects we were working on, either existing products that we wanted to optimize the routes from because they suffered from low yield or high cost, or new targets that we wanted to access. And it was really important to have those chemists who were working on the projects be able to use the software themselves to design the routes so that they could customize it based on their needs or their limitations in their lab. So for instance, if there were certain reactions or reagents they needed to avoid for safety reasons, they could add that information. They can also define, what starting materials they have available and give it some of that input as to what reactions they want to do or avoid. Since this was an experiment, we did have some rules associated with it. So any of the routes had to go back to commercially available starting materials that either already were in the lab or that were available to ship that week, because once they got back to the US, they just had 8 weeks to execute on the routes. So kind of trying to mimic a project timeline there. And then the other major rule was that no substantive changes were allowed to the routes. So they really had to do the steps as proposed by Cynthia, those steps in that order. But they did expect to do some optimization of reaction conditions. So you'll see Cynthia does give you a good starting point in terms of some typical reaction conditions for all of the routes that it proposes, but it doesn't necessarily give you the most optimized conditions for those particular substrates. So they did expect to do some optimization in that way. I won't go through all the results, but I do want to give you one story here. So this compound alpha hydroxyatizolam, which is a metabolite of the drug atizolam used to treat depression. This is one that came from our reference materials site that they wanted to add as a new product. There was need for this as a reference material, but this was actually a novel molecule, so there was no chemical synthesis of it. And really the challenging piece of making this molecule is the installation of this hydroxyethyl group here. And so when the chemists were originally planning a route to make this molecule, they actually proposed doing this installation at a very late stage in the synthesis. This being the high risk step, that was basically deemed too high risk. The reference materials site primarily is mainly a production site, so they had kind of limited resources for R&D, and they thought that that route would likely require too much R&D, and so it was not executed on. But they then chose this as a good target for the Cynthia evaluation to see if they could use the software to find a more viable route. And so if we look at the output we initially get from Cynthia is this graphical node representation, where we have this yellow circle here, that's going to be the target that you put in, so the molecule you're looking to make. Any of these pink circles, these are commercially available starting materials. Any of these purple circles, those are actually representing unknown molecules, and where we have this light blue halo around some of the molecules is indicating you need a protecting group at that step. So of course, if we actually look at the chemistry here, what was proposed overall was an 8-step synthesis, including the protection and deprotection steps. And what was really key about this synthesis was this 3-component Jewald reaction being done at the very beginning. And this would be able to install that protected hydroxy group in the right position as it's forming the thiophene ring. And so this was, really important to do that high risk step up front, because then they would know very quickly if the route was going to be successful or not. And in fact, they were able to isolate that thiophene product in a 30% yield. This was enough to take on for the rest of the synthesis. And from there was more standard benzodiazepine forming chemistry, and then finally making that triazole ring to isolate the final product in a 3.2% overall yield. This is of course a modest yield, but was a significant result since this was a compound that didn't have any published synthesis and now allowed us to add that as a new product in the portfolio. We also were able to access additional analogs using parts of the route, and it represented a 40% cost savings over that route that had originally been proposed by the chemist. So really a significant result for us. Another feature in Cynthia is that it actually has the ability in some cases to be able to predict potential side products that may form. And this is one that we actually were also able to verify using this experiment. So we noticed with that 3-component Jewald reaction, a significant amount of a side product that we were able to identify with spectral data as the result of this Novanagle condensation. And in fact, this was a side product that Cynthia had correctly predicted could form as a result of this reaction. So another sort of helpful piece of Cynthia to help you identify side products or have ideas of, impurity profiles or things like that. So, I mentioned that we put 7 targets into the experiment, and I think going into it expected maybe a 50% success rate, but in fact, we actually ended up with a 100% success rate. So all 7 of those targets, the routes proposed by Cynthia, were able to be successfully executed in the lab, and in many cases led to really significant cost or yield savings over the original routes. And so we were able to publish the results of this experiment in this Chem publication that came out in 2018. This is an open access article, so if you're interested in seeing more of the details, you can find that there. But I have kind of a quick summary here of, some of the, like I said, costs and yield savings we found with some of these routes. So for instance, for this compound here, we were actually able to do a chromatography-free synthesis with the route found by Cynthia, which of course significantly improved the yield. Another one, this hydroxylorazidone here, was a compound where the only published route was under patent, and we were able to find a patent non-infringing route with Cynthia, and that's actually kind of another way that you can customize the software. If there are certain routes that you want to avoid, you can mark key disconnections and tell Cynthia not to make cuts there. And so, overall, this was a very successful result. We were able to, increase yield and reduce costs for some of our existing products, find efficient syntheses for new compounds, and you're also able to generate new IP or even protect your IP space using Cynthia. And so, of course, as a result of this very successful evaluation, we did acquire the software back in 2017, and we are continuing to develop it and now have it available for commercial licensing. So just to quickly wrap up, we've been able to show that Cynthia really is the first tool to deliver computer-aided design that has successfully been validated in the lab. It's been able to save chemists time in planning and execution. This is, of course, a tool to augment your expertise as a chemist. So, another tool in your toolbox to help you do your job even more effectively. And this is really becoming the chemist's preferred starting point in route design and planning. And as we have more and more chemists using the tool, we're able to use that feedback to help improve it. And we are really in a very end user focused development phase now. And so with that, I think probably before we jump into the live demo, I think there's a poll that we're going to do quickly.
There is, yes, I will get this poll ready to go. So this is for us to get to know a little bit more about you as an audience. So what do you think would be the most important benefit to you of using a computer-aided retrosynthetic design tool such as Cynthia, of course? Do you think it would be most useful to help you find greener chemistry? So that's where you can use it to find the routes that don't involve harmful compounds, or perhaps to minimize some of the side products that you might otherwise get? Is it for IP protection? You want to make sure that you're doing this in a way that protects your own intellectual property? Do you want to save time and money by finding a route that perhaps uses cheaper initial resources or fewer steps? Do you want to avoid hazardous chemistry? So we already mentioned so reach restricted compounds there. Do you want to find routes to produce your molecules that do not involve dangerous compounds? Or is it a little bit of all of them? Is it all of the above? And I'm just going to watch as people vote. I can see lots of people have been asking us really good questions. Do keep those questions coming in. There's a questions box almost certainly at the bottom of the GoToWebinar panel. I say almost certainly because you can move that panel around and cut out the different boxes and so on, but it's probably down at the bottom there. And you can ask any questions. We've got quite a few really good questions in already, but do keep them coming because we do keep a record of them, so we will be able to pass them on to Emma. We also have Lindsay Rickershauser, who is also from Millipore, who is helping us out to answer some of your questions. So if her name pops up in response to your question, then that's why, and thank you, Lindsay, for joining us. I think that's just about enough time for people to vote. So what I'll do is I will share those results, and we can see that there's a lot of people who are being quite mercenary about this. It's about saving time and money by finding more efficient synthesis routes. But most people think it's a little bit of everything. They think that they can benefit from all of the different things that having computer-aided retrosynthetic design is there to do. So I mean, Emma, clearly you guys saw the potential to achieve all of these things in Chematica, and I'm assuming you would also have ticked all of the above yourselves.
Yes, I think so. Like I said, sometimes, different projects have maybe more or, different priorities where it's more important to, protect IP or maybe avoid a patented route, or, maybe you're really specifically looking to find a less hazardous route. But I would say overall, in general, we're probably looking for, all of those things.
Excellent. Well, thank you to everybody for completing the poll. I can see more of these questions coming in. That's great. But now we're moving on to see the real thing in action, aren't we? So I will hand control back over to you, Emma, and let's see Cynthia in action.
Great. Are you seeing my screen again? Sorry.
Yep, we've got you again now.
Okay, perfect. Great, so then this is what our Synthia web app looks like. So we first launched this web app version commercially about a year ago, I guess maybe a little over a year ago now, in March of last year. So it is a web app that you access using a web browser. Every user has a unique login and password that they use to log in, and then it stores all of your searches, so you can see, some past searches I've run here. We do have different options that you can organize them in different folders to make them easy to come back in and look through later. But for this, I do want to walk you through what goes into setting up a search. So we hit this new analysis button, and there's actually a few different ways that you can use Synthia. So our main mode is this automatic retrosynthesis, and this is primarily what I've been talking about, where you can put in the target that you're looking to make, you give it some criteria of what's important to you for the synthesis, and then Synthia will plan those full step-by-step routes back to commercially available starting materials. But we also have a manual mode, And this is kind of more of a hands-on approach. It uses our same database of hand-coded chemistry rules to determine what transformations are possible. But now instead of planning complete routes, what it does is shows you all of the retrosynthetic cuts available, just going one step back. So this can be really useful for idea generation or just understanding, different ways you could approach making that molecule. And then you can choose any of those routes that you like or any of those reactions and expand those nodes further to sort of manually build a route that way. For this, I'll go ahead and just choose the automatic retrosynthesis. And then in terms of putting in your target, we do have a known database in Cynthia. So if you're looking for a known molecule, you can search the database by name or by a CAS number. But we also have a molecule editor. So we do have a Marvin sketch editor in Cynthia that you can draw any structure here or paste one that you've drawn before. So I'll go ahead and paste a structure in here. This is also where if there's stereochemistry in your molecule, you can define the stereochemistry, and Cynthia will look for routes that either are starting from chiral building blocks or that are, enantioselective syntheses. So we'll take that into account. This is also a place where, like I said, if there were certain routes you were trying to avoid, or there's a known route and you're looking for something different than that, you could mark certain bonds in here and tell Cynthia not to make cuts at those points. But for this, I'll just kind of leave it as is. And once you're happy with the molecule, you can hit confirm and configure. And this is where you can now tell Cynthia, what's important to you and really customize those search criteria. And we have a lot of different options for that. But we do also just have this quick start. So this is usually a good place to start if you want to just get a quick idea of the results Cynthia will get with the default parameters. And then it's very easy to rerun a search and change some things to kind of narrow in more on what you're looking for. So there's different areas that you can go through that we've kind of split out in terms of customizing the search conditions. This first page here are some filters that you can add that change a little bit how this how the search works or what it's looking for. So for example, we have an option if there are heterocycles in your molecule and you specifically want to build those from scratch, you could choose this option to cut all heterocycles. And now, even if that's a heterocycle that's commercially available, it will show you ways to make it from non-heterocyclic building blocks. But the main area where you really tell Cynthia the criteria you want it to use to rank the paths is here in the scoring functions that I mentioned during the presentation. And so if you were to hit the quick start and just use the default conditions, what Cynthia would consider are going to be the number of steps in the route, the cost of starting materials, and the number of protecting groups are kind of the main criteria. And it gives sort of a balanced weight to each of these, but you can adjust these. So for instance, if you actually don't really care that much about the cost of starting materials and you just wanted to find the shortest routes, you could check this box here, and then it doesn't give very much weight to the cost relative to the other things. Similarly, there's options to sort of set your tolerance for using protecting groups if you want to try to find a protecting group free synthesis, or if you don't mind using them, you can choose the different options here. And then you can also direct Cynthia toward more convergent or more linear paths. By default, it will show you a combination of both. And then this middle section here is where you can really kind of fine tune it, where you can tell Cynthia if there are any specific structures, so whether they're starting materials or intermediates that you don't want to use, you can put those in here. And again, we have another Marvin sketch editor, so you can draw structures in, and then Cynthia won't use those in the routes. You can do the same thing with keywords, and this is where you can avoid whole reagents or kinds of reactions. And so if there's less environmentally friendly reagents that you want to avoid, you can add those in here. And then you can also make whole lists of molecules. And so we do actually have some lists that are preloaded in Cynthia of some regulated substances. And so if those are ones you want to avoid, you can just check those options here, but you can also make custom lists. And so if you wanted to include, for instance, a list of all of the reach-regulated compounds or something like that, you could make a list, and then every time you just click that option, and then Cynthia won't use any of the molecules on that list in your synthesis. And then finally, we have the stop conditions over here, and this is where you tell Cynthia what you want to use as starting materials. And so we have a few databases in Cynthia. We have the commercial compounds that will show up as these pink nodes. These are coming from our Sigma Aldrich catalog, but it does also include our Building Blocks Explorer and our Aldrich Market Select portfolio. And so altogether, that contains around 4.2 million substances coming from around 80 different vendors. And we have price information for all of those. So you can set a price threshold for your search. By default, it's going to consider anything that's $1,000 per gram or less, but you can set that lower or higher depending on your preferences there. We also have a database of known compounds that will show up as these kind of light green nodes, and these are now compounds that have just been published in literature. We have a database of around 10 million substances, and here you can set a popularity threshold, and this is now a minimum popularity which refers to the number of times that compound has been published in our database. And so again, the default is set at 15, so we'll only consider something a starting material for your synthesis if it's been published at least 15 times. But you can set that higher or lower. You can actually turn off any of the databases altogether if you just wanted it to go to the commercial compounds. And then we also have the ability as an add-on for your license, if you wanted to include your internal inventory as starting materials in Cynthia, you can do that as well. And that could be any commercial compounds that you just have in-house or intermediates that you've made, and that can really further customize the results you see that are going to be different from what anyone else would see in Cynthia because you have a unique database there. Cynthia will show you the top 50 paths that it finds. That's the maximum number, but you could set that lower. And then you can also define kind of how complex of a search you want it to run. We have, again, some default options that we've set based on what are typically required for kind of different levels of molecules. For most of these small to medium complexity molecules, the standard analysis is fine. For a more complex molecule, something with a lot of stereo centers or a larger natural product or something like that, you may want the long analysis, and you can actually customize it further. I'll go ahead and stick with this. This really defines how much of that graph of possibilities that Cynthia is going to search through, and it will also affect the time that the search takes to run. Typically, for kind of a small to medium complexity molecule with a standard analysis, it will usually finish in around 5 to 10 minutes, but you can actually start to see results as soon as they form. For longer searches or for more complex molecules, it can run as long as a few hours or even overnight. But again, you don't necessarily have to sit around and wait for your results in Cynthia. You can set up a search and go do something else, and you'll get an email when your search is done. So I'm going to go ahead and hit start on this just to kind of show you how that works. So the computation will start running. You can actually track the status over here in this computations tab. It will actually send the search to our servers where it goes into a queue, and as soon as it gets picked up, then the search will start running, and so you can kind of follow that there. So you can see how long the search is going, and then as soon as it starts finding results, they will automatically load here. But like I said, we don't necessarily have to watch it run, so this is one that I've run before, so I'll go ahead and jump back home and open up those results. And so just for reference, we can check this particular search finished in just under 6 minutes with kind of the, basically standard parameters. So Cynthia will show you the paths here, like I said, with these kind of colored nodes. We do have a legend in the tool, so you know what those represent. So this yellow target here, that's what you're looking to make, and all of these pink circles, those are commercially available compounds, and you can see the price per gram underneath. This is based on purchasing basically research quantity scales, so you know, buying in gram quantities, so you can kind of take that into account when you're considering looking at the prices there. But we can, instead of just looking at those colored circles, you know, as chemists, we typically like to look at the structures there. And so we can turn on the structures, and then they still have those colored boxes, so you can see what, you know, if it's commercially available or a known compound, and still the price pretty quickly. In terms of looking through your results, you can click on any of these reaction nodes and it will show you the reaction that's being proposed there. And then if we click on these details, it gives you some additional details about the reaction. So all of the reactions will show you some typical reaction conditions here, as well as some illustrative references. And so these are things that are hand coded as part of the rules of looking at, you know, all of the chemistry precedent for this kind of reaction and choosing some, you know, conditions here, as well as then selecting a few references that are really representative of that kind of chemistry. And these are direct links. So if I click on that, it will open up the DOI of that paper so that you have that as some precedence of where this reaction is coming from. You can see there's sometimes, you know, a few different options. A lot of times they'll try to choose a seminal paper for that kind of chemistry. Sometimes the use of that reaction in a total synthesis, or a methods paper where there's a significant substrate scope, something like that. So you do have an idea of, like I said, the literature precedent for that chemistry. It's not necessarily going to be the references that are going to be that reaction on substrates that are the most similar to what's being proposed here, but we do have the option to look into our known database to see some similar reactions, where it will now look to see if the same chemistry has been done on similar substrates, and if so, it will show them here with a similarity score. So you get an idea of other reactions that have been done that are similar to this. In terms of looking through your results, I mentioned Cynthia will give you the top 50 paths that it finds. You can, of course, go through them one by one. Just scrolling through here, it does rank them with this path score. So you see the lowest scoring path first. That's basically the best one based on that criteria you gave it. But we do have some filters, so you can filter them further. You can set a specific number of reactions in the path, or filter them based on if they have regulated substances or protecting groups. But we also have this very useful group by family filter that basically is a similarity. So if you're looking for kind of the most diverse options in that path, I can set that down lower. And then for this particular result set, when I set it at 20%, I'm down from now, instead of 50 paths, just the top 13, but these should be kind of the most diverse options. The other option we have to look through the entire result set is this graph mode, and this is now showing me all 50 of those paths in a single view. So you get a really quick visual of the diversity of the result set, of, you know, how many last disconnections there are to get to your target, and where there are some key intermediates that you know, have multiple ways to make them. We can, you know, zoom in and out of here, and we have, you know, the same nodes here. You can still click on any of those to see what the structure is. But the other thing that we can do is more holistically look through the reactions by generating this reaction report. And this is now going to list all of the reactions in the graph over here on the left-hand side. And there's different options to sort through them. So by default, it's showing me all of the last steps first. So if I want to just look through those, I can click on any of these reactions and it will highlight it in yellow over here if I want to kind of follow the rest of the path. But there's other options to sort it. So for instance, if there's a stereocenter or a ring in your molecule and you just want to see the reactions that are installing those features, you can sort it that way, or alphabetically by reaction name if there's specific chemistry you're looking for, and then it will group all of the same chemistry together. So this is where you can see if, you know, the same or similar reactions are being done in multiple paths. You can click on those here and it will highlight them in the graph. And then similarly, we do have a molecule list that's now going to show all of the molecules present in the graph. And again, you can click on any of these. apologize, sometimes it just has to get refreshed quickly while I've got a search running. Is if I click on any of these molecules in the graph, it now highlights them in yellow, so I can see where that reaction or where that molecule is being used, or maybe what paths aren't using that, and I can focus on some of those. And then also for any of these commercially available compounds, we do have direct links to our website, so you can see, you know, specific pricing for that particular product. And then the last thing, I did mention that you can, you know, it automatically saves all of your results in your account that you can go back in and look at later, but you can also export results as a PDF. So if there are a few paths that you wanted to save, you can do that and generate a PDF where it has sort of the overview of the path, and then goes through each reaction and gives you all of that same information that was in the reaction details, the typical conditions, the references, any starting materials, and protecting group information if it has that. And so with that is really kind of all of the main features that I wanted to show you with Cynthia. So I think we probably have some time now. I'd be happy to answer some questions.
Thank you very much, Emma. I'm always surprised. I've seen Synthia in action a few times, and it obviously contains a lot of information. It's a very practical tool, but I'm always surprised how aesthetically pleasing those sort of spider web reaction charts really are. It's really quite a beautiful tool as well as a very helpful one. We've had a lot of questions, and we probably won't get time to get through all of them, but we have got a record of everybody's questions. So what we'll do is we will pass these on to Emma and Lindsay afterwards, and if there is an answer that they can provide to you, they will be more than happy to do so. But with that in mind, Emma, lots of people are asking similar things about where can we get the software from? How much does it cost? Does it run in the cloud? What sort of computer does it need? Where is a good place for them to go to find out those sorts of answers?
Yeah, that's a great question. So you can go to our website, so millporsigma.com/Cynthia, to be able to find more information. And if you're interested in, you know, purchasing or talking to someone on our team, there's, you know, a little button that you can click on there. I believe that says, you know, to get more information, and then that will, you know, send your query to our team, and we'll reach out and we can do, you know, a private demo and show you more about, you know, how it works, and talk about, you know, pricing and different options. We do have kind of a variety of licensing packages that are very customized, you know, for users, so we can talk about that. And we also have a lot of other, you know, resources on our website as well. So that's a good place to find that.
So let's dive into some of the audience questions. Aref would like to know what the main advantages of Cynthia are compared to similar tools like SciFinder or Reaxys.
That's a great question that always comes up, of course. And, you know, what we like to talk about is really the unique things that are about Cynthia. So, you know, we really are the only retrosynthesis tool that has a hand-coded rules database. And, you know, I talked in the presentation about the advantages of that, of really being able to, it allows it to look at the full molecular context of, you know, the reaction as it's planning it, and allows us, you know, a more comprehensive coverage of the reaction space. So that's something that's really unique about Cynthia. We are also, I believe, the only tool that so far has a published, you know, bench validation, where we have actually tested it and shown that the routes planned are, you know, can be successfully executed in the lab. You know, some other things that about Cynthia, we do have really a lot of different ways to customize the tool. You know, I kind of quickly pointed out a few of them, but there's really even more than that, in terms of, you know, ways you can really tailor it to make sure that you're getting results that are the most relevant to you. And that's something that's, I think, a little bit different about Cynthia than maybe some of the other tools.
Excellent. Well, lots of people had asked that, so thank you very much. Let's okay, so you talked about one of the advantages of these being hand coded, but Shota would like to know if you're going to apply this program to a novel machine learning system. So do we want to take it out of being hand coded, or in addition to that have a machine learning approach?
Yeah, that's another great question. And certainly, like I said, machine learning is a very, powerful tool. And we actually are looking at ways that we can incorporate aspects of machine learning into Cynthia. So, still kind of maintaining our core of the hand-coded rules, but being able to use that as kind of additional filters or things like that. So we are exploring ways that we can incorporate machine learning into Cynthia as well.
I guess another place machine learning could come in handy would be to keep an eye on the literature. You talked about using machine learning to strip rules from the literature, but it could also just be used to flag up to you when you may need to review the rules for any given path or any given reaction.
Yeah, that's a good idea. Yeah, of keeping track of that. I'm not sure if that's something that we're doing, but that would certainly be a useful application.
So Tito would like to know, is there a limit to the molecular complexity that Cynthia can handle?
Yeah, that's a great question. Not really. You do have to draw a discrete molecule, so we don't really have a way to, if you have a large polymer of just kind of a specific molecular weight range, you can't necessarily do that. But I don't think there's any real limit other than, just, you have to draw it in the sketch editor. It does have to be, like I said, a single specific molecule, so there's not a way to kind of designate just an R group, for instance, or like I said, a molecular weight polymer. But we can, otherwise kind of handle any molecule that way. And we actually do have an option, if I'll just maybe expand on that a little bit,
Yeah, sure.
as I mentioned with having an R group there, is we do actually have a batch mode. So you can't necessarily specify, one group that's different, but you can upload a whole list of molecules to kind of set up a search at once. So if there was kind of a few different, positions that you were substituting somewhere, you could do something like that.
So although you can't fully wildcard, you don't have to just go through and do everything manually one at a time. You do have some time-saving steps in there.
Right.
Both Chris and Martin, and I'll be honest, the rate at which Chris needs to come in, we may well have had a few other people asking the same question. Does Cynthia consider reactions with enzymes? So is enzyme catalysis something that you can look at in the pathways that you choose?
So we do have a handful of rules in the database related to enzyme catalysis, but it is not an area we have extensively covered right now. The challenge with that is the enzymes are usually very substrate specific, so it's difficult to kind of have them in, the general way that we do with Cynthia, where it can be applied to, substances that it hasn't specifically been published before. So we don't have a ton of that, but we, like I said, have a few of those reaction rules are related to enzyme catalysis.
Thank you very much, Chris and Martin, and anybody else who asked a question about enzyme catalysis. We still got a few minutes. This might be a little bit philosophical, but Rio has said, what do you think will be the role of organic chemists once Cynthia or similar software could find all of the needed routes without any human help?
Yeah, well, I think the good news for us as chemists is that I think we are far away from really the software being able to completely do it without us. So I did kind of mention this at the end of the presentation, that we certainly don't think that Cynthia is something that can replace a chemist. You really still need the chemist to kind of drive it and, customize it, and then look at the results and decide, what reactions I actually want to use, or what I want to do there. So I don't think that we're, really replacing a chemist. We're just kind of evolving, how you're doing your job. So, getting away from maybe spending, a lot of time doing maybe some of the more tedious work, or planning this out and being able to very quickly now have a lot of options of routes that you can go through, and then spending more time doing other things, or being more creative. I think our jobs will always kind of evolve to use our brains and be able to have input there, but this is just something that will help us do that.
So it's a valuable new tool for chemists, but certainly not a replacement for them.
Certainly not yet, and I'm not sure if ever. I think we'll probably still have a role, but we'll just kind of evolve as we have better tools to work with. Just my opinion. Yeah.
Okay. Well, as I said, it's a philosophical one there, but edging perhaps away from the point of today's webinar. Maybe we should do a chemistry philosophy webinar at some point, and automation would almost certainly hit the decks in that one. We've got 2 minutes left. Let's have or this hopefully a fairly quick one. Can Cynthia be used to design small molecule drugs and ligands which are predicted through bioinformatics? That's from Erytrojoy.
That is an interesting question. So, I mean, it can certainly be used to synthesize, ligands or small molecule drugs. It doesn't, I guess I'm not sure what the question is exactly. If you want to link it directly to your kind of bioinformatics software, we don't kind of have a direct link that way, but that's something that we may be able to develop in the future, to be kind of able to have it all sort of talk to all of your cheminformatics and bioinformatics tools, and that's where like I said, we do have that batch mode, so you could upload kind of a list of possible molecules that maybe you got from a molecular modeling software, and then be able to run them quickly together in Cynthia.
I think we've technically run out of time, but let's just have one last question. We've talked about IP a lot, and you did say that it's possible to put your own proprietary structures in. Does Merck get to see, do you guys get to see everything that people put in there, or once you're using Cynthia, is it all safely enveloped away in your own system?
Yeah, that's a good question. So yes, everything that you put into Cynthia is securely stored, so we aren't able to see any of the searches that you're running, and we have a lot of data security in place as well to make sure that nobody else using Cynthia is seeing that. And like I said, even within your own account, every individual user has a unique login and password, and only you have access to the searches that are in there.
Excellent. So rest assured that anything proprietary that goes in is still for your eyes only. Well, thank you, Emma. I'm sorry that we've overrun very slightly, but thank you to everybody for all of your questions. I think there have been hundreds of questions that have come in the last few minutes alone. So we will certainly send all of those over to Emma and Lindsay, and if they can get back to you, then they will. But I think this is a good sign, because we do have more webinars coming up arranged with Millipore Sigma. So keep an eye on chemistryworld.com/webinars, and you'll see the other things coming up where you can ask more questions of our fantastic guests. And thank you again to Millipore for arranging Emma to come and join us today. Thank you for suggesting the topic, and for getting Lindsay involved to help answer your questions in the background here as well. We will send everybody who attended a certificate as a way of saying thank you. We'll also send you a copy of the recorded webinar. So if there's anything that you missed or you just want to see again, that will be in your inbox soon. So thank you ever so much once again for joining us today, and a big thank you to Emma. Thank you to Millipore Sigma. I'm Ben Varsler from Chemistry World, and we'll see you in the next Chemistry World webinar. Thanks again.